So we're going to talk about cell division. We're actually going to talk about cell division two different ways. This particular conversation is going to be about mitosis. Later on, we're going to talk about meiosis, and they do very, very different things. So this cell division talk is going to be on mitosis. We're going to do it in a couple of parts. So I'll have this lecture, and then I'll have another lecture following up. So I wanted to start out with first just thinking about this, which is why do cells actually divide? There's multiple reasons why cells divide, and it depends a little bit on whether you're multicellular or unicellular. So for us, for multicellular creatures like us, we are doing cell division for two main functions. We're doing mitosis for replacement of damaged cells, so repair, and then we're doing it for growth. You all start out as a single cell, and so you have to divide that single cell into the trillions and trillions of cells that make you up now. If you are a single-celled organism, say like a prokaryotic organism like a bacteria, you will use cell division for reproduction. So it's a way to reproduce asexually. And bacteria do this all the time, and you can see here bacteria with their single loop of DNA will simply copy their DNA, separate those two copies, and form two new cells. So it's a way of reproducing. It's another form of cloning. I wanted to spend some time talking about how eukaryotes do this mitosis process. First, though, I wanted to talk to you a little bit about how the DNA is packaged, because one of the major things we have to do in mitosis is make copies of the DNA and then separate those copies into the two new cells. So we have to make two cells that are genetically identical. And how this works is a function of how the DNA is stored in the cell. So we've talked about DNA looking like this, a double helix. And in one of the previous lectures, when we talked about gene expression, I talked to you about the idea that the DNA is actually wrapped around these proteins called histones. Histones are important in terms of storing DNA. Think about going on a trip. If you go to pack for a trip, there's a right way and a wrong way. If you want to pack for the trip, the wrong way, and maybe the more fun way, is to take all your clothes, throw them in the suitcase, and then jump up and down on the lid of the suitcase until you can zip it shut. The right way is to fold up those clothes, and that totally makes sense. You can fit more clothes in a smaller space. If you think about the DNA in your cell, we talk about it being this double-stranded molecule. It's very long. Each human cell has 6 feet, 2 meters of DNA in it. So you have to get this very, very long strand packed up into the nucleus. So the way that gets accomplished is the DNA gets wrapped around these histone proteins. So we'll coil it up, and it's a way of kind of folding and organizing. The DNA then gets coiled up into this thing called a solenoid, which is a coil of coils, and that's the way the DNA is stored. It's actually stored in these loops. Now, when we go to copy the DNA, we'll make 2 copies here, and this is what you're seeing, but usually when you think about chromosomes, you usually think about this X shape. That is where the DNA has actually been supercoiled. So we coil it, and then we coil it, and then we coil it again. So it's wrapping around itself again and again and again, and you get this X shape. And what you're actually seeing here are these 2 chromosomes, this single chromosome that's made of 2 parts. Now, I wanted to talk about that. So we often draw these chromosomes in X's, and when you see a picture like that, what you're actually looking at are 2 copies. So these 2 things are called sister chromatids. They are copies of each other. So if I look at these blue ones here, this chromosome is the exact genetic copy of this chromosome. They have the same genes on them. Same thing here, this red one over here, this sister chromatid has the same genes as this one. Now, there's another sort of term here that I want you to think about, and that's homologous chromosomes. So if you think about human beings, and you're probably aware of this, if you didn't remember, we have 46 chromosomes in each one of our cells. We have 23 pairs. Where did those pairs come from? Yeah, your mom and your dad. So if you think about it this way, if you think about the blue chromosome being from your mom, the red one being from your dad, you have pairs of chromosomes. You have 23 pairs. In each one of those pairs, you got one from your mom and one from your dad. So this is this thing called being diploid, having two sets of chromosomes. So when you see chromosomes like this, and they never actually look like this in the cell, this is where the chromosome has not been copied yet. This is where the chromosome has been copied. So we have to do some work before we can actually do cell division. So I want to talk about that. Now, one way we think about cells and how they do their thing is we think about this thing called the cell cycle. This is a bit like the life cycle of the cell. So if you think about your life cycle, right? Usually when I ask students in class, they say, oh, you're born, then you die, and that's a little bit oversimplistic and a little harsh, actually, right? You're born, you grow, you become an infant first, and then a toddler, and then you become a tweenager and a teenager, and then you go through that awkward puberty thing, and then you become an adult, and then you become an older adult, and then you die, right? So you go through all these things. Cells do the same thing, and one way we can represent that is this thing called the cell cycle. So this represents the cycle of the cell, and I'm going to draw this up here. It's a little awkward, but we're going to do this anyway. So if you think about it here, I am going to start with a cell right here. So I'm going to draw this cell. There's my kind of crappy looking cell. So this is a cell undergoing the cell cycle. And it's going to do this thing. This part of the cell cycle here, that's in yellow, red, and green, this is what's known as interphase. Interphase consists of these three different parts. One called G1, one called S, and one called G2. Now the G here stands for gap, but I always tell students, think of the word growth. So what's going on here in G1 is the cell is growing because we've just divided it. So we had this big cell and we've divided it. It has to get bigger. So by the time it gets over here to the end of interphase, it's going to be a much larger cell. So what's going to happen is G1, the cell is going to grow. The organelles are going to make copies of themselves. The cell's just going to get bigger. Now, the S part of interphase is what's known as synthesis phase. That's where the DNA gets copied. So we have all this work to do. We've got to grow, then we have to copy the DNA. And then in G2 or gap2, we're going to continue to grow. And the cell will just do its job. So we have to do all that before we get to mitosis. So there's a lot going on here. Now, the idea of mitosis is I'm going to take the two copies of DNA that I have, and I'm going to separate them. Now, you're probably familiar with the stages of mitosis, but I wanted to go through them again, right? So they are prophase, metaphase, anaphase, telophase. Now, interphase, which we just talked about, interphase is actually the longest part of the cell cycle. You'll notice here that this is kind of short, the mitosis part. In fact, most of your cells aren't dividing all the time. Your cells actually only divide as an adult maybe 10 to 30 times during your lifetime. So most of the time your cells are in interphase and not dividing. Well, since we're running a little bit long on time in this video, I'm going to do a separate video where we talk about the stages of mitosis. So we'll do that next.